just going to put the chat up. Carbohydrates. What are the 4 macromolecules besides carbohydrates? So you've got carbohydrates. What else do we have? You can type it in the chat. What are the macromolecules? Protein, good. Lipids, excellent. And a starch is a carbohydrate. Yes, so we've got our carbohydrates. And what is DNA made of? What kinds of acids? The nucleic. You may or may not know that, but yeah, so there's basically 4 kinds of macromolecules that we require. So you might recognize where your carbohydrates are. Well, polysaccharides of carbohydrates are things like starch, and you find those in your fruits and vegetables. Another polysaccharide, saccharide means sugar. Carbohydrates are sugars. They're simple sugars and complex sugars. So the polysaccharides, I think mostly people call the long chains complex. So I just call them polysaccharides. So cellulose is a polysaccharide. That is the structure of a plant cell wall. And you need that. You need that for fiber. Yeah, but we don't store our sugar as cellulose ourselves. We store it as glycogen. So glycogen is the polysaccharide that we store in our livers and our muscles and other places for energy eventually. Yeah. So some things to note about these different molecules are, what are their properties? Are they hydrophilic? Are they hydrophobic? How do they function in our bodies? So this one is hydrophilic. A carbohydrate is hydrophilic. The general formula is Ch2O. And we're going to put a little N there because if the N is 6, it's glucose, a 6-carbon sugar, C6H12O6. So the monomers of carbohydrates are monosaccharides. Monosaccharides. They're the simplest. The general formula is C6H12O6. And they are found as chains, but also as rings. So in liquid, they tend to be rings. And as different structures like that, they have different types of sweetness even. So the 3 major monosaccharides are glucose, galactose, and fructose. They're mainly produced by the digestion of complex carbohydrates that we eat. Interestingly, all of these monosaccharides have exactly the same molecular formula. The differences are their structural isomers, their structural shape. So this is glucose, which comes from cane sugar, galactose, which comes from milk, and fructose, which comes from fruits. Disaccharides, di means 2. So disaccharides are pairs of monosaccharides. So there's one that you eat all the time, that's at your table, sucrose. So sucrose is a combination of glucose and fructose. Lactose is a combination of glucose and galactose. And maltose is a combination of glucose and glucose. And so you might be wondering, what are you expected to remember at this point? Well, I wouldn't expect you to remember these kinds of diagrams and tell the difference between sucrose, lactose, and maltose. However, you should remember, because of its importance in diet and in structural and different functions of the body, that the disaccharides are sucrose, lactose, and maltose, and what they consist of. So that you should remember that. You should remember that Monosaccharides, the general formula is C6H12O6, and that there are three, and the names of the three. Yeah, so structural pictures like that, I don't expect you to reproduce. Occasionally, if I need you to remember one, or if I think one is important enough in its shape to recall or have in your knowledge bank, I'll let you know. Polysaccharides are such things as starch, cellulose, and glycogen. So these are long chains of glucose, and they're formed by dehydration synthesis. Starch is produced by plants, and we digest starch with amylase. And we start digesting starch immediately in our mouths. Cellulose gives structure to plants, but it's also fiber in our diets. So we don't digest cellulose. There are some bacteria that can digest cellulose, and they are in the guts of some animals, like cows, for example. But for ourselves, the fiber in your diet is necessary to abrade the walls of the digestive system and to help food to move through the digestive system. Glycogen is our energy storage polysaccharide. It's produced by animals. Our liver cells do the work. They synthesize glycogen after you eat, and that takes the glucose out of your blood. So it maintains your glucose levels in your blood. This is a chain of glycogen. It's branched. So what are the functions of carbohydrates? Well, there's different kinds of carbohydrate functions, and carbohydrates make friends with different kinds of other kinds of molecules, and they're known as conjugated carbohydrates. So what are their functions? I think I'll add a page here. What is the main function, would you say, of carbohydrates if you were to make a guess? So you can type that in the chat. The main function of carbohydrate. What do we use it for? Excellent. Energy. It's our source of energy. Used in cellular respiration in the mitochondria to give you ATP. But there are some conjugated carbohydrates that you may or may not have heard of. One of them is known as a glycolipid, glycolipid. And they are an external surface of the cell membrane. Yeah, so this moiety, there's two moieties here. One of them is a lipid, and the other one is a carbohydrate. There are also, pals, glycoproteins. Glycoproteins. They are also the external surface, on the external surface of the cell membrane. And they provide a function such as mucus. So they are what's known as the mucus, or the mucous membrane of the respiratory tract, for example, and digestive tract. So they provide lubrication, and in the respiratory tract, the trapping of particles. Another conjugated molecule is known as a proteoglycan. That is also a protein moiety and a carbohydrate moiety, but the carbohydrate part is dominant. And it functions in cell adhesion. So cells that make up your tissue, they work together. So the muscle cells, for example, are lined up beside each other and they work in concert for a single contraction of a muscle. So the cells need to adhere to one another very tightly. Also, proteoglycans are that gelatinous material in your tissues. So it makes up things such as the vitreous humor in your eyes, and it lubricates joints also. So when there are different molecules in a macromolecule, they're called moieties. Moieties of macromolecules. It's a chemically different component of a conjugated macromolecule. Proteoglycans, for example, have the protein moiety and the carbohydrate moiety. Okay, here is an unscramble. So what I'd like you to do is unmute, and look at these for a little while. I'll give you about a minute or so to look at the words and see if you can unscramble them and let me know what they are in a minute. I'll ask everybody what they are. All right. What's the first one?
Macromolecule.
Nice. What's the second one?
Protein.
Protein. Excellent. The third one?
Lipid.
Lipid. Nice. The fourth one?
Carbohydrates.
Carbohydrate. Nice. The fifth one?
DNA.
DNA. The sixth one?
Sugar.
Sugar. Nice. Yes. The seventh one?
Fat.
Fat. The eighth one?
Polypeptide.
Polypeptides. Yeah, that was a hard one. The ninth one?
Nutrients.
Nutrient. Good. And the 10th one?
Starch.
Starch. Right on. Okay, well done. Well done. So that brings us to the end of carbohydrates. Let's look at the next macromolecule of interest, lipids. Lipids are hydrophobic. So if you ever put oil in water, you know it's going to clump together. Doesn't like water. Hydrophobic means water hating. Yeah, it's an organic molecule, of course. It has a carbon backbone. It's less oxidized than carbohydrates, so it has more calories per gram. So what that means is that it has more, what does that mean anyway? It has more hydroxyls, I believe. Yeah. So, don't quote me on that, I'm not sure. When you oxidize something, it means you take away the electrons and the hydrogens. So yeah, it must have more hydrogens. So these are the types of lipids that we're going to look at. The phospholipids, which are the bilayer of every cell in your body, animal fats, and vegetable oils. So there's 5 primary types of lipids in your body. 5 types. Let's write those down. There are fatty acids, which you may have heard of, triglycerides, phospholipids, eicosanoids, those are signalers, and steroids. So those are the 5 types that we'll look at. Fatty acids, triglycerides, phospholipids, eicosanoids, and steroids. So let's look at fatty acids. They're a chain of 4 to 24 carbon atoms. There's a carboxyl group at one end, that's this end here, sorry, this end here, and a methyl group on the other end. A methyl group is simply a carbon with 3 hydrogen atoms attached to it. They're polymers of 2 carbon acetyl groups. So this group right here is a 2 carbon acetyl group, and they're all strung together. Fatty acids. So you might have heard the term saturated and unsaturated. A saturated fatty acid is one that has carbon atoms completely saturated with hydrogen. So for example, stearic acid is saturated in that every carbon molecule here in the chain has a hydrogen atom attached to it. There are no double bonds of carbons attached to other carbons. However, in an unsaturated fatty acid, there is at least one carbon atom double bonded to another carbon atom. And you might think, well, what is the significance of that? The significance of that is the unsaturated kind with the double bonds tend to bend. And so when they bend, they can't stack up against other ones, and they form a liquid because they can't be solid. So if this were, these are all saturated, well, they can easily line up beside each other, become solid at room temperature. So lard, an animal fat, is solid at room temperature because it's saturated. The unsaturated ones will have a bend like that. And they won't line up and stack neatly together. Instead, they move around. So they're moving around here. And that makes them a liquid at room temperature. A triglyceride is a combination of fatty acids bonded to a glycerol. This is a glycerol group here. As all synthesis happens, dehydration synthesis. In this case, our triglycerol is a combination of palmitic acid, a saturated one, stearic acid, a saturated one, and linoleic acid, an unsaturated fatty acid. So this is going to be an oil. Interestingly, the saturated fatty acids are more likely to be converted to cholesterol. And the unsaturated fatty acids are not. And so that's why having a lot of saturated fatty acids can lead to an overabundance of cholesterol in the body. So this is your glycerol, your 3 fatty acids. You have the addition of water. And with the addition of water, with the taking away the dehydration of water, you've joined these 3 fatty acids together to the glycerol. And here is the little oxygen in between that's bonded them all together. Triglycerides are called neutral fats because they're the fatty acids. They're fatty acids. They have a carboxyl end. That's what makes them acids. But this is the carboxyl end on this end, and now it's bonded to the glycerol, so there's no longer an acid present. So that's why triglycerols are known as neutral fats. To hydrolyze them or to break them down or digest them, an enzyme is required known as lipase. So enzymes are nice because they're, for naming anyways, often they're named after what they digest. And so a lipase digests lipids, particular triglycerides. So at room temperature, a triglyceride is, if it's a liquid, it's called an oil. Polyunsaturated fats from plants. If it's solid, it's known as a fat. These are saturated fats from animals. The function is energy storage. So if you use up all of your carbohydrates, your next source of energy will be your fats, your triglycerides that are stored. But also, fats provide insulation, particularly around vital organs, and subcutaneously to keep your body warm, and shock absorption for organs as well. So maybe some of you, hopefully we'll get to do some dissections, and you'll see that the eye, for example, is completely covered in fat. It protects it from hitting the eye socket, which is bone. Another kind of lipid is a phospholipid. It's amphiphilic in character. That means it's both hydrophobic and hydrophilic. It has a hydrophobic tail, very similar to neutral fats, with 2 fatty acids, though, not 3, and a hydrophilic head, which differs because the 3rd fatty acid is replaced with a phosphate group. And what do we know about phosphate groups? Do they have a charge? Do phosphate groups have a charge? Yes, yes, they do. And that makes them hydrophilic. That negative charge causes them to be easily dissolvable in water because water is polar. So let's look at a phospholipid. This is lecithin, for example. So this chain is saturated, this chain is unsaturated. That makes it sort of a semi-liquid. It has a nitrogen-containing group called choline. This is the phosphate group with a negative charge, the glycerol group, and 2 fatty acids. So you often see phospholipids drawn like this. So this whole region is drawn as a circle. That's your hydrophilic head, that region, but it includes the choline and the phosphate group. The hydrophobic region includes the glycerol and the fatty acids. So phospholipids are a major component of cells. So you have your, I'm just drawing them straight, although they're not straight. It's just faster. It's often drawn like this. This is a phospholipid bilayer. So this makes up the plasma membrane of every cell in your body. And the reason it works is that these, the heads face out towards the water of your body. So they easily remain in water. But the tails, which are hydrophobic, they clump together. So these can spontaneously form. And that's why they function so well as a plasma membrane. And we're going to talk a lot about the plasma membrane when we get to cells. But they are liquid and always moving around. And as such, they leave space for things to move through. So eicosanoids are derived from fatty acid. They're chemical signals, so they function as chemical signals. Prostaglandins is one of the really important ones. Its role includes inflammation, so signaling for inflammation and blood clotting, hormone action, labor contractions, and control of blood vessel diameter. Steroids include cholesterol, and all other steroids in the body are derived from cholesterol. They're all cholesterol is what they're synthesized from. There's some things are added. So things like cortisol, progesterone, estrogens, testosterone, and bile acids, they are all steroids. Steroids are extremely important for the functioning of our body's endocrine system. Cholesterol is required for proper nervous system function, and it's a very important component of cell membranes. Only animals produce cholesterol. 85% we produce ourselves, and we get 15% from our diet. So just going back to our plasma membrane, say this is cholesterol here, and here, and here. Cholesterol has this odd but fantastic function in the cell membrane in that the cell membrane is largely liquid. So if it gets very warm, the molecules move around more and they could dissolve and disintegrate. But the cholesterol holds them together when temperatures get a little bit warm. If temperatures were to get cold, then the phospholipids would stack closer together and make it more difficult for things to pass through. The cholesterol, however, keeps the phospholipids from stacking together too tightly. So cholesterol is a very important part of the plasma membrane. You may have heard of such things as lipoproteins, and some people say cholesterol is so bad for you, but of course you need quite a lot of cholesterol for various functions in the body. However, you can have an overabundance of cholesterol. And the way cholesterols, you might be wondering, how are cholesterols transported around the body or even other lipids? Because our blood is mostly water. And so if you had different fats and cholesterol in the blood, they would just clump up. So you need to transport them by surrounding them with proteins, which are hydrophilic. And in the case of cholesterol is transported by lipoproteins. And there are such a thing known as low-density lipoproteins and high-density lipoproteins. And the low-density lipoproteins are, I'm going to say, bad. So they're not so great. The high-density ones are better. And what that means, a low-density lipoprotein, is that there's a high ratio of lipid to protein. So low-density protein, loads of lipid. High-density lipoprotein is the other way around. There's a low ratio of lipid to protein. And so the high-density lipoproteins help to transport cholesterol to the liver, back to the liver, to take it out of harm's way and to get it out of the blood, for example. So it's good to have a high proportion of high-density lipoproteins. How do you get them? Well, interestingly, you can get them from not smoking, from exercise, and from maintaining body weight. So having too much cholesterol and excess will cause more low-density lipoproteins to form. Cholesterol, though, is required for so many functions in the body, and all steroids are derived from cholesterol. So this is a structure that I think you should remember. Put a little star beside it, because it's quite recognizable. Cholesterol has 4 rings, 1, 2, 3, 4, and functional groups, hydroxyl groups and methyl groups. Yeah, so whenever you see rings like this, you assume that every corner has a carbon. So the corners have carbon. So this is a very handy table that you could look at some point. It gives you the functions of different lipids. So the fatty acid functions, they're the precursors of triglycerides and a source of energy, for example. Triglycerides are also insulators as fat, a filling space for organs such as between your eye and your eye socket. They bind organs together and they're cushioning. Phospholipids are the major component of cell membranes and they aid in fat digestion. Eicosanoids are your chemical messengers. We didn't talk about fat-soluble vitamins too much, but vitamins that can dissolve in fat are very important for blood clotting, wounds, vision, of course, and calcium absorption. Cholesterol is a component of cell membranes and a precursor of other steroids. Steroid hormones, they are not coming up, but the steroid hormones that are derived from cholesterol are chemical messengers between cells in the endocrine system. Bile acids are steroids that aid in fat digestion and nutrient absorption. So a bile acid is kind of like a surfactant. So it can help digestion of fats, which otherwise would clump up. Let's look at, what time is it now? We're doing well. Oh, we're doing very well. Okay. I'd like to look at proteins, and then we will stop the lecture. Proteins, organic molecules, they're polymers of amino acids. Amino acids, there's 20. There's 20 amino acids. They're all identical except for this R group. So all amino acids have an amino group, they all have a carboxyl group, and they all have a hydrogen bonded to those other 3 electrons in the outer shell of the carbon. Yeah, so the property then, because they're the same in every other way, is determined by the R group. And the amino acids determine a protein's structure and function. Which amino acids are present and in which order. Very important, in which order. So what are amino acids? They're organic molecules. This is an example of some R groups you might find in different amino acids. This is methionine. Methionine is always the first starting amino acid in a chain of a peptide. A cysteine, tyrosine, and arginine. So they vary a little bit in their properties. In particular, some are non-polar, like methionine and tyrosine, but some are polar. Proteins that contain very many amino acids are, as a result, often amphiphilic, both hydrophilic and hydrophobic. So amino acids. Well, just the same as our other macromolecules, we can synthesize proteins by dehydration synthesis. To get a dipeptide, we take 2 amino acids and remove a water molecule. The bond is called a peptide bond. And the structure is a dipeptide, di meaning 2. There are also other peptides called tripeptides. They have 3 amino acids, of course. There's oligopeptides. They have between, let's see, 10 to 15 or so. Polypeptides. They have more than 15. And I'm running out of room. Proteins have generally greater than 100 amino acids. So when a protein is synthesized, it goes through several stages during synthesis. The first stage is putting together the primary structure, in other words, the order of amino acids. So that's known as the primary structure. It's the order of amino acids. The secondary structure occurs because this protein is forming in water. It starts to fold or wind and form hydrogen bonds. So here are the hydrogen bonds shown here and here. And it's those hydrogen bonds that cause this string of amino acids to start to form a shape. And sometimes the shape is known as a beta pleated sheet, and sometimes it's known as an alpha helix. And you'll have both when the full protein starts to form, which is known as the tertiary structure. So the tertiary structure is the functional form of the protein, and sometimes the quaternary structure, some proteins have more than one unit. They have different subunits. So 2 or more subunits. Insulin is a very simple structure. It's a protein, it's a hormone, it's composed of 2 polypeptide genes, and they're attached by these disulfide bridges, which are very strong and maintain the shape of insulin. Proteins also make pals with other things. Conjugated proteins, they may contain a non-amino acid moiety, sometimes known as a prosthetic group. One that we've looked at before is hemoglobin. Hemoglobin has 4 polypeptide chains, and each chain has that complex iron-containing ring known as a heme moiety. And that is where the iron resides, and that is where the oxygen attaches. So here's a picture of hemoglobin. Remember, we saw that in our video. And these are the heme groups in the center of each subunit peptide. So one thing about proteins which make them so functional in the body is that they have a particular shape. So the 3-dimensional shape of proteins gives them their function. And we'll talk about functions in a moment. But it all depends on that shape. They can change shape. So an important property, they can change shape. So for example, you might have a plasma membrane here, and you have a channel protein of a certain shape, but it doesn't let anything through unless it manages to change shape. So in this case, it's closed. In this case, it's open because it's changed shape. So this is an example of, say, opening and closing membrane pores. If it's an extremely drastic change, say in extremely hot temperatures or extremely pH environments, so proteins are made in particular environments of a particular pH, a particular temperature. For us, it's 37 degrees. So if it gets really, really hot, the proteins will start to denature beyond being able to function. So that's why a fever, if it gets too high, can be quite dangerous. However, we have fevers in order to try and denature our whatever is affecting us, like viruses or bacteria. And that kind of denaturation can be permanent. So let's look at protein functions. These are just some examples. Here is collagen. Is a long fibrous protein. It functions as structure. Some proteins are enzymes. Some proteins are channels. That's the one we just saw. Closed channel, open channel. And some proteins are receptors. So, functions, structure. Maintaining the structure of parts of the body. So, some proteins involved in that are collagen. And you might have heard of keratin. Keratin is a protein you find in your nails and your hair. So those are examples of proteins that provide us with structure. Communication is also a function of proteins. So hormones, for example, some hormones are proteins, some hormones are not. Some hormones are lipids or steroids. Also, cell receptors are receptive to signals. That makes them a communication molecule. Membrane transport is another function. So proteins form channels, for example. And proteins, as we saw in the lipoproteins, can also be carriers through the blood of lipids. Another function is catalysis. And we saw that enzymes function as catalysts. They function as recognition and protection. So antibodies are proteins. Some proteins function in clotting. And some are antigens. So antigens, they're generally actually glycoproteins. And they are like identifier cells. Or not identifier cells, but identifier proteins. They identify your cells as belonging to yourself. Yeah, some cells don't have that. Sperm doesn't have that. So sperm has to be separate from the blood, otherwise your immune system of the male would destroy the sperm. Another function of proteins is movement. So skeletal muscle cells and other muscle cells, cardiac and smooth muscle, the movement is due to actin and myosin. Muscle contraction due to actin, which is one kind of protein, and myosin, which is another. Another function is cell adhesion. Proteins that bind cells together. So those are the main functions of proteins. Let's take a quick look at enzymes. So in a moment, I'm going to get you to do an exercise, a crossword puzzle, using a lot of these terms. So enzymes are catalysts. A catalyst promotes reactions. In this case, rapid reaction rates. Sure, even spontaneous reactions in the body would happen anyway. That's true. But they would happen so slowly because the molecules aren't meeting each other very often that nothing would ever happen. So they function as catalysts. Some terminology: the substrate is the substance an enzyme acts upon. And enzymes are often named for their substrate with an A's added. Like amylase digests as a type the type of starch known as amylose. What enzymes do is lower activation energy. So molecules are moving around, but they're not bumping into each other very much. So the enzymes bring them together, and they provide an environment conducive to a reaction. So here is an enzyme process that is going on. This is the substrate, in this case sucrose. That's what the enzyme will act on. The substrate will land at an area known as the active site. So this is the active site. And it will bring that sucrose into an environment that makes it more likely to split. So in this case, water is added, and the products, it's split here by hydrolysis, and the products fructose and glucose are released. So in this case, with the active site, it's making the bonds between these two monomers weaker and easier to break. So that's the job of the enzyme. Cofactors are non-protein partners of enzymes that are required for enzyme function. Iron, for example, copper, zinc, magnesium, calcium, they change its shape and therefore create an active site. Yeah, so lots of enzymes can't function at all without cofactors. So there's a difference between cofactors and coenzymes. Cofactors are minerals, coenzymes are organic. They're usually derived from water-soluble vitamins. Yeah, so some of them are pantothenic acid, for example, which is in coenzyme A. It's required for the synthesis of triglycerides and ATP. A niacin, a coenzyme, an NAD, and riboflavin, and FAD. So these are all functioning in cellular respiration. They're very important for that process. For example, here is NAD. So NAD attracts the electrons towards itself. So glucose in this case is split because some of those hydrogens and electrons are stripped from it. In other words, it's oxidized. And in this case, so glucose is oxidized. Electrons taken away. And what happens in this case is overall in aerobic respiration, we're going to synthesize ATP. So we really need that coenzyme for NAD. So I'd like to talk about oxidation and reduction because that's just one of the molecules, some of the molecules. I don't really like that slide though. So yeah, I'm going to skip over it for now. I'd like to talk about oxidation and reduction because it's extremely important to cellular respiration, which is where we get our energy. So we said before that we can pair chemical reactions. One is exergonic and one is endergonic, and that's awesome. So oxidation is exergonic and reduction is endergonic. You can think of it this way. Oil rig. Oxidation is loss of electrons, and reduction is gain of electrons. So in this case, we've oxidized glucose and we've reduced NAD. And now NAD has a couple of, one extra hydrogen along with the electron. So the chemical reactions in the body don't happen necessarily as just one thing turns into another. Rather, there are intermediate steps, a chain of reactions. So this is a reactant, this is a product, and these are intermediates. And in this case, our reaction is going in this direction. And these are enzymes. This is one enzyme and another and another. So how is it that we can stop a reaction from carrying on? Well, one way is to deactivate the enzyme. So the end product, D in this case, may deactivate. Sorry, that's not the enzyme, hang on. D may deactivate one or more of the enzymes because we have plenty of the product. All right, I am going to stop there.